So we're going to start talking or reviewing the leaf. Do you remember that from grade 11? Let's draw pictures of leaves in grade 11. Cross sections, that is. So we're just going to draw a little leaf and remind ourselves how leaves work, because of course photosynthesis, what we've been talking about, occurs primarily in leaves, right? Not entirely. There are other parts of the plant that do contain chloroplasts and are green, but their functions are often quite different. So the leaf is the main photosynthesis factory. Okay, so let's start by drawing a little group of cells like this across. Actually, no, my mistake. Let's start by doing them sideways. Let's draw some cells sort of this way. Okay, like this. Just little cells all in a row, right across the top of our picture. Like this. This, of course, would be the top of the leaf. So we're looking at a leaf edge on. We're looking at the very, very thin edge of a leaf in cross-section under a microscope to see the detail that we want to see. Okay? To make them look like cells, we can throw a little nuclei inside there in various spots. I colored them green on purpose because they contain chlorophyll. These cells, they're chlorophyll-containing cells. And this is called the epidermal layer of the leaf. Epidermal layer. I don't know if I'm going to be able to fit all the words on this picture, so what we'll do is we'll draw the picture first, and I'll give you the names. You can write them down as we go, and we'll try and summarize them after to make sure you have them all. Underneath the epidermal layer is another layer of cells which sort of is oriented differently, something like this. This kind of orientation is called a palisade. Picket fences are sometimes made this way. A palisade is an old word that comes from fortresses that were built by stacking up logs vertically like this and making a wall, a palisade. So this is called the palisade layer. If you put your nuclei in, don't put them all in a line. Give them a little bit of variation and it looks more real. If you put them all in the same line, it'll look like cells. It's amazing how that works. Okay. Then we have the next layer. I'm going to slow down because I know you're all panicked. Well, I can do something simple while I'm waiting. On top of this, I'll do it in a sort of a kind of a browny color. Maybe this orangey color or this color. It doesn't really matter. There's a layer on here of wax. It's a cuticle, they call it. It's basically a layer of wax. You'll remember that waxes are forms of lipids, right? And the leaf secretes this wax, of course, to keep the water out and in, keep water in more than out. But it's a waterproofing layer. It also helps to deter insects that want to chew on this leaf, so it has some defense and protection type thing. And it's just basically a wax. And you can often see it on plants. If you ever see a leaf and you see raindrop, the water drop hits the leaf and balls up and rolls off. And of course, that's because waxes are fats and they are hydrophobic. They're non-polar. So it causes the water to ball up and roll off rather than soak into the leaf. But that's really not what the leaf wants. The leaf is concerned about water that's in the leaf getting out. It wants to maintain the water inside. So it waterproofs the leaf. Okay, the next section is what we call the mesophyll layer. So we have the waxy cuticle, the epidermal layer, the palisade layer, and then the mesophyll, which is also green and has chlorophyll and is photosynthetic. These cells are sort of bundled all up in blobs, but the key thing here is that there are some spaces in here. There are air spaces. So we'll draw a few like this. Leave a space sort of in the middle here, because we want to use that later. Okay, and they're just in sort of groups, oblong cells, something like this. And they form a sort of a spongy type texture or structure. So they're often referred to as the spongy mesophyll. Okay, I'll put sort of something here, down here like this, there, the spongy mesophyll. And of course, we could put some little nuclei in there to make them look like cells. And then we have a bottom layer. I just go down a little. A bottom layer, very much like the top layer, the bottom epidermal layer, which would go across the bottom. But watch this. I'm going to leave a little space in a few spots. That's very important to leave the space, because the bottom of this leaf actually has tiny holes that can be regulated by the cells on either side. And these holes are, of course, very important because this is how a cell breathes, or a leaf breathes. This is where gas exchange goes in and out of the leaf. Okay. Now, the next part, or the last part of this, would be the actual vein of the leaf. If we're doing a cross section, the vein will appear like the open end of a pipe, right, in this cross section. And so we have to draw ourselves a couple of different little circles. So I'm going to pick a little blue color here. And what we have is some little tiny blue pipes. These little pipes are all bundled together, and they are the xylem tissue, which transport water through the cell. And then I'm going to use a red color, and I'm going to put in underneath it a bundle of other tissue. These are called the phloem tissue. So the bundle has two different kinds of tubes in it. The xylem for transporting water, and the phloem, which transports sugars through the sap. And the direction of flow is what's important here. Typically, the xylem is coming towards the leaf. It's bringing water from the roots up the plant to get to the leaves, because water is needed for photosynthesis, right? Remember, water, we have to pull the electrons off of water to get the whole photosystems to work. And then the sugars that are made by the leaf in the process of photosynthesis are then pumped through the phloem cells out towards the rest of the plant for whatever purposes it needs, right? Sometimes those sugars are stored elsewhere. They're pumped to the roots. Some plants store their starches in roots rather than in the leaves. Other plants store them right directly in the leaves. But nonetheless, all of the cells need sugars. So at some point, the phloem will bring that out. Okay, now, this forms what's called a vascular bundle. It's got the xylem and the phloem in the vascular bundle. Then there are, I'm going to use a different color here, maybe a purpley color, I guess we'll use, for brown color. Let's use this brown color. There are little cells sort of around here that hold all this together. And these cells are going to be very important in what we're going to discuss later. They're called bundle sheath cells. Bundle sheath cells. And these bundle sheath cells basically form the bundle sheath. They hold all the vascular tissue together in a bundle. The bundle sheath cells. And then, of course, around that, there would be other mesophyll cells. So these mesophyll cells that I've drawn already would sort of be crowded around here as well. Now, I'm only drawing one to show the picture. But these vascular bundles, they branch all through, like blood vessels in a way. So here's one that would be large, but they tend to branch all over. So all of these mesophyll cells that I have here would be very close to a vascular bundle, either a larger or smaller one. But we're only drawing one just because we want to make the picture simple. So just like your bloodstream, you know, we think about an artery or a vein, but there are tiny, tiny blood vessels just about everywhere in your almost every cell in your body is directly beside a tiny capillary or blood vessel. It's kind of the same thing in the leaf. These vascular bundles stretch and branch out through the leaf, but we're just drawing one for the time being. There should be another waxy cuticle along the bottom. So let's get that brown-orange color and go along the bottom here with some more wax. Don't cover the hole, though. Otherwise, the hole won't work. There's the wax on the bottom, protecting the leaf. Okay. Now, in grade 11, you hopefully went through this. Everybody's familiar with this picture, right, I think? You've all seen it. Okay, so you kind of understand what everything does. Our focus is going to be that vascular bundle in the middle, those mesophyll cells around it, and the bundle sheath cells. Because what we're going to find is that there's a couple different kinds of plants that use slightly different ways of doing photosynthesis. And the reason is because they live in extreme environments. Plants have adapted to live all over the place. But to understand that, we have to go back and review how the gases are exchanged in a plant. So these holes, which are called the stomates, or the stomata, if you use the Latin, are controlled by the two cells on either side. Maybe what I'll do is color in the two cells on either side to distinguish them. Let's use my green crayon here and just color in these dark cells. They're called guard cells. Okay? And just to remind you how guard cells work, basically they're sort of attached together at the ends, kind of like two sausages in a sausage link, but at both ends. So I'm going to go down below here and just talk about guard cells a little bit, just separate from the picture. So what you have is a guard cell. Oops, let's go green. You have a guard cell that looks something like this. And the other guard cell is sort of attached to it, something like this. And we get the stomate, or the stomata, the hole. The stomate is the hole in the middle. So this is the hole. We're looking at it sort of flat on instead of cross-section now. And these two cells are attached. Okay? So I'll just shade them in. Now, the cells I've drawn are full of water. And so like a water balloon, when water gets into the cell, it causes the cell to expand. And so these cells will expand, but because they're attached right here at the ends, when they expand, they push out and they open this large hole in the middle, the stomata. So the stomata is open when there's tons of water available to the plant, when the water is the plant is well watered. If the plant begins to lose water, the water balloons on either side, the guard cells, will begin to lose their water and get less sort of firm. They'll get all flaccid and limp. And so what happens is in conditions that are dry, they tend to sort of limp up. And when they limp up, the hole between them closes. So we have conditions of wet conditions and dry conditions. And that's basically how the guard cells work. So those holes can be regulated. Well, here's the thing. Let's go back to our leaf now. It's very important for where photosynthesis is happening inside the mesophyll. It's very important for us to get carbon dioxide gas into the leaf because we know that CO2 is important for photosynthesis. So typically, the stomates want to be open to allow carbon dioxide in. What's one of the products? What's the gaseous product of photosynthesis? What gas does it make? Oxygen. And so at the same time, there's oxygen that's building up inside here. Whoops, let me use my smaller line. There's oxygen building up inside here because of photosynthesis, and that O2 has to get out of the cell, right? So having the stomates open is a good idea because essentially it means the leaf can breathe. It can bring in new fresh CO2 from the outside and get rid of the oxygen that's being built up from photosynthesis. But there's a huge problem. With these holes open, something else gets out. Precious water that the leaf needs. It needs it for photosynthesis. It's going to leak out of these holes because typically, the inside of a leaf is very humid and wet. So compared to the outside air, which is usually a little bit drier, there would be a constant flow of water out of the stomates. So the plant has a problem. It wants to open the stomates to bring the CO2 in and get the oxygen out, but as long as the stomates are open, it's losing water. This water loss is actually called something. Do you remember the big word it's called? Transpiration. And leaves lose a ton of water to transpiration. They pump up tons of water through their roots, but a lot of that water is lost to the air. And they don't have a choice, because if they close the stomates to save the water, they suffocate. They don't get to exchange the gases they need. So the plant has to play a little game. And what's happened is that plants have evolved to live in very humid climates and also very dry climates. And so what they've done is they've made some small modifications over the years of evolution that basically give us sort of three different ways of doing it, of doing photosynthesis and living in conditions that are dry. So in other words, how do we make sure we get carbon dioxide in when we live in a desert, like a cactus, for instance, but not lose our water? Because in a desert, water loss would be fast and immense, right, with the dry air. Whereas in a rainforest, where it's much more humid, it's not so big of a problem. So the opening and closing of the stomates is how the plant sort of breathes, but has this water loss problem. And so the next thing we're going to do is we're going to learn about three different sort of strategies. Well, one we already know. The standard photosynthesis that we've learned, that's done as one option, and that's for plants that are living in conditions where there's enough water vapor in the air so it's not a problem. But the other plants that live in the deserts or dry conditions, they have to have an alternative way to solve that problem. So we're going to stop this video, which is leaf review, and then we're going to talk about how the bundle sheath and the vascular bundle adapts to the dry conditions.